recording in progress. There we go. So thank you very much for attending tonight. We just sort of finished up with our chick chat, which honestly is discussion, right? Chatting, coming up, talking about babies, talking about master's projects, talking about pigs or sheep or goats or whatever it is that we feel like talking about. We have one more upcoming online session on October 28th. We'll be learning about heroes to hives, so some beekeeping plans. And then we're super excited about our in-person Pearls of Production coming up on November 8th. So real quick technical stuff. I'm going to go ahead and drop this. Sorry about that. I'm going to drop this in the chat one more time. My information, just in case you need to reach out to me during tonight. I am tech support for today. So my email is bromfieldc@missouri.edu. My phone is 813-624-8196. So I'm going to go ahead and mute myself when I'm done talking, so that way if phone calls come through or if text messages come through, I'll be able to respond to them and not interrupt the presentation. If we have questions that come through to me, I may unmute to let you know that there are questions. But at this point, we've got pretty much everybody muted other than our presenters. So ask us questions. If you have questions, somebody else may also have the same question, and you'll be doing everyone a favor if you help us to clarify. Feel free to type it in the chat, raise your hand so you can click the hand raise button, or unmute and ask us. We have special thanks to Beringer Ingelheim, who has helped defray the cost of tonight's presentation. We also have special thanks to FCS Financial, as well as, I believe, Missouri Cattle Women's Cattle Production.
Yeah, Cattlemen's.
Okay, Cattlemen's, as well as Missouri Pork Association. So tonight we have Dr. Andrea Pitkin, who is going to talk about some gut health issues. Let's talk about SRP technology. I've got one more slide before I'm going to go ahead and turn it over to Heather. Heather, who is our 2025 chair. So our committee members are here. We've got a handful of the people online available who will be able to answer some questions if you have additional questions. But Heather, would you like to take it away and introduce Dr. Pitkin?
Yeah, so I met Dr. Andrea Pitkin during the Passion for Pigs conference. Was that just last year or the year before that? Was that just last year?
December.
Yeah, just this last year. She did a phenomenal job explaining this topic that could be kind of confusing. Does just a phenomenal job. And I was like, our ladies at Pearls really need to listen to Andrea. So Dr. Andrea Pitkin is a 2011 graduate of the University of Minnesota College of Veterinary Medicine. Prior to obtaining her DVM, she completed a master's program in infectious disease in 2009, in which her thesis was based on biosecurity principles, focusing on understanding the spread and prevention of PRRS virus into swine herds. Dr. Pitkin has worked in practice, multiplication, research, and the pharma industry, most recently with Vaxxinova US Worthington, since June of 2022. She resides in Watertown, South Dakota with her husband, Paul, and their 3 boys, Marcus, Conrad, and Brant. So, Dr. Pitkin, I will turn it over to you so that we can hear this exciting topic. It's really a lot of fun.
I'm going to go ahead and get those slides up.
Perfect. So, while you're doing that, I just want to take the opportunity to say truly thank you very much. 2 of my favorite things to talk about are pigs and quite frankly, how much my children annoy me. I think all mothers can relate to I love you, but you tend to drive me crazy, right? And honestly, sometimes I feel the same way about pigs. They are magnificent, amazing creatures, and sometimes too smart for their own good. So kind of like I say, pigs and children are my 2 favorite things.
Well, I almost have it up.
Okay.
Of course,
Perfect.
I'm the one having the tech issues tonight.
Okay. I'm glad the tech person is having the tech issues. This makes me feel very, very good about myself when I have them.
All right. Are you seeing the presentation?
I am. I
Okay.
am.
Go ahead and give me the heads up when you want me to turn to the next slide.
Absolutely. So thank you for doing that for me. So, again, super excited to be here and talk to you about essentially what we're going to call do you have gut health issues? If you have pigs, you're very familiar with the term scours, right? And scours is literally that dirty word on a pig farm. We don't want to see them. We don't want to experience that with our pigs through any stage of production. And I can absolutely tell you, if you're experiencing any type of gut health issues, you're not alone. This is a common conversation that we have with clients and customers, and I'm excited to tell you about a vaccine that Vaxxinova has to help specifically with E. coli and salmonella gut health issues. So excited to tell you and learn more. Next slide, please. So before we really get into anything, there's a couple key points that I want to make first, so that while we're talking through this presentation, hopefully this helps make a little bit more sense to understand why we think SRP proteins make a good vaccine target. So SRP proteins are well recognized by the immune system. So when you're looking at vaccine technologies, if your vaccine technology targets a protein versus a carbohydrate or sugar versus a fat, that's kind of the key order. So if you can get a vaccine against a protein on a particular pathogen, the immune system recognizes that the best. So it gives you a very robust immune response. The other thing about SRP proteins is that they are highly conserved. So what that means is, and I'll give this with salmonella as the example. These SRPs that we target on salmonella are conserved across every kind of salmonella. So these proteins, since they're so key in helping E. coli and salmonella survive, it's hence why they've been conserved across these different types of bacteria for many, many years. We figure out a way to attack that, because since they're so conserved and they're on lots of different coli variants, salmonella variants, it makes it really good, again, target, because the immune system responds, and we only need a few isolates because they're carried across different bacterias. And I'll explain that more in just a little bit. The last thing I'm going to leave you with before I explain all of this is the process that we use to manufacture SRP is simply a fermentation process. It's a growing process, right? So we grow this bacteria for the vaccine in an environment to get those SRPs to express. And we're very proud. This is not anything that we use to brag, but we're extremely proud that we've actually made over 1 billion doses of this vaccine across different species. I mean, a billion. That's a massive number. So tell you that to let you know this has been around for a long time. It's a growing process to get that vaccine or to get that bacteria to grow and express in its normal environment. So it's a very safe, very well used, well tested, smooth vaccine. So next slide, please. So you keep hearing me say this term SRP, and what the heck does that mean, and why do we really care? So SRP, and not that I would expect you to remember this after I say it, just simply call it SRP because it's a heck of a lot easier, stands for siderophore receptor protein. And as I was just saying from the previous slide, siderophore receptor proteins are these protein molecules that are conserved across different E. coli's and across different salmonellas. So let's talk about what this thing actually is and why we care about it. Next slide. So in extremely simple terms, we look at these SR proteins as a doorway, as an air valve. So you can all recognize that that's a football, a basketball, and a volleyball, right? All those balls need air to function. You can't dribble a flat basketball. You can't serve or set a flat volleyball. Now, maybe if you're Tom Brady, you can throw a flat football, but that's neither here nor there. But all these balls need air to function, right? So they all have an air valve. So think of that as a critical access point to that ball that they have to get air in them to function, okay? So keep in mind this concept when we talk about SRPs. So this is kind of the high level, gets you thinking about how this functions. So if you would go to the next slide, please. Here's the technical version of that. So SRPs are a type of what we call porins. Porins are channels across a bacterial cell wall. So again, go back to that air valve, right? That air valve goes across from the outside to the inside of that ball so you can get air in. These porins, which are a type of protein, are what allow different molecules to get in and out of a cell. So a cell needs water to come in and out of it to maintain its function and homeostasis. It needs nutrients, right? All bacteria need iron to survive. So they're going to have a porin, or again, this type of protein that allows iron to get into the cell because they need that as food. They need that as fuel. Okay. So again, similar to that air valve. Again, think of that as a way for that iron to get in and out. So that bacteria needs iron, just like that ball needs air. And the reason we give these examples with different types of ball is if you look at that basketball as an E. coli, you look at that volleyball as a salmonella, the football might be a Klebsiella. That's really important to our dairy friends, right? All those, if you look at them as different bacterias, have that same air valve. It's the exact same concept. The Klebsiella, the E. coli, salmonella, all have that air valve or that porin, specifically that siderophore receptor porin, which is going to bring in iron into the cell. Okay? So I really want to take the time to pause here and say any questions, because it's important to grasp this concept that air valve, that porin, is the same across different bacteria, and that its function as an SRP is to bring that food, that iron, into the cell so it can grow, it can function, it can colonize, it can implement virulence. Any questions on that from anybody? And I love questions. Please ask them. I hate feeling like I'm just like talking at people, right? Like, truly ask me questions.
I'm not seeing any questions in the chat, but definitely thank you for explaining that, because it makes a lot of sense knowing that this is a piece that is on lots of bacteria.
Mm-hmm. Yep. Keep that all in mind.
And I do want to highlight what you said, that this isn't, while this talk is specifically looking at pigs, this technology is used in all sectors, like you were talking about the dairy friends, right?
Yep.
Poultry, they use, I mean, it's across all species, not just swine.
Correct. Yep. And I have a couple slides on that later just to show some of that data. But yeah. All right. Next slide. So if you've talked to a nutritionist, a veterinarian, a production specialist, you've probably heard of villi in the gut lumen. So if you think of the intestine as one very, very long tube, right? Inside that intestinal tube are these structures called villi. So that's the pink I'm blanking on a word. The pink blips that you see across the bottom of the screen. Those are the villi reaching up into the lumen. And here in this animation, we've unfortunately got some E. coli molecules floating around. Those are the guys that have the little tails on them. They're called flagella. That's how they move and work their way through the gut. So they're in there and they're going, you know, I think I'd really like to colonize and cause an infection. So what they tend to do, and as you see in the animation right now, they're diving down into those villi. They don't want to necessarily be up in the lumen. There's no place for them to attach. So they want to go down into what we call the crypts or the deeper parts so that they can attach onto the villi walls or the gut walls, set themselves up to grow, to multiply, to again colonize and cause infection. So when those E. coli are up roaming around in the top of the gut or in that lumen, they're really not doing much. It's again when they dive down, that's where they want to set up shop and you know, again, hang out, get themselves attached to the gut wall and start to implement their virulence factors. So next slide, please. So again, going back to what is an SRP and why do we care? SRPs are an iron acquisition mechanism that bacteria have. So when they go down into the lumen, is the animation not working on this slide? Can you possibly, oh, if you go back. Oh, maybe you skipped one here. There we go. There we go. Sorry, we were ahead of slide. So SRPs are these doorways on the bacterial cell wall for iron. So what this is showing is a zoomed in image of the E. coli, and it's sending out that blue spiky molecule because it's trying to grab iron from the host, bring it to the E. coli or the salmonella, because that's its food, that's its fuel, that's what drives it. So that SRP, again, is that doorway or that opening that allows iron to come in. So now, next slide, please. All right, so here we go. So you can see that blue spiky molecule coming away from the E. coli. It goes and grabs one of those maroon round balls that's depicting iron, and it literally brings it back to the E. coli and goes inside the cell, drops the iron off, and then it actually recirculates back out into the gut to grab more iron and bring it back to the E. coli. So it's actually kind of a ingenious little mechanism, right? That when that E. coli goes down into those crypts, as I was showing on that slide with the villi, there's not a lot of iron down there. So it's turning on this iron stealing process to go grab iron where it wants to be down in that crypt and still find fuel, find that food that it needs to colonize and implement its virulence. Okay? So why do we really care about this? Next slide, please. So if you look at these three different sections of pig intestine, an unhealthy pig might have a little bit of colonization, a little bit of disruption on the gut wall. So if you look at that top piece of section that says unhealthy pig, you can see the reddish pink areas. That's nice, healthy gut tissue. And you see a little bit of that brown. That's necrosis from E. coli colonizing the gut. So if you get too much of that, going all the way down to that bottom picture, if those villi are all colonized over and not able to absorb nutrients, what happens to our pigs when they can't absorb nutrients? They don't grow very well, right? They don't want to eat because their gut hurts. They're not going to grow very well because not only are they not going to eat well, then they're not going to absorb nutrients. They're not going to absorb water if their intestine looks like this. So those pigs that you get in your barn that just look like they eat and eat and eat and they don't put any weight on, it's likely because they've had damage to their intestinal tract. And this can be caused not only by E. coli, you can see this from salmonella. Salmonella typically causes more damage more down in the colon or large intestine. And that's actually where you absorb a lot of your water. So if you get damage up in your small intestine, like E. coli likes to do, you don't absorb nutrients. Damage later on, you don't absorb as much water. So you can imagine how if rotavirus has come in and blunted off all your villi, now E. coli comes in and colonizes it and really destroys it. Salmonella comes in and doesn't allow these pigs to absorb water. You can see why you constantly have diarrhea or issues in your herd or pigs not growing, because they just don't have the nutrient absorption capacity anymore from how these pathogens can impact their gut. So the whole point of this vaccine is trying to understand, can we stop this process before it even starts? How can we prevent E. coli and salmonella from even attaching and doing their damage to the gut wall? Next slide, please. So when animals are vaccinated with SRP vaccine, essentially what they are doing is creating an antibody response. And those antibodies, as you can see in this animation, recognize those porins, those holes, those SRPs on the surface of the E. coli and go, I'm just going to block you because I found you. I'm going to attach to you. You don't need to function. Like, that's literally what's going on here is, okay, they recognize the protein, like what I was talking about initially, right? Because that's a good vaccine target. So these antibodies go and literally create a barrier so that when that bacteria is sending out those siderophores, those blue spiky molecules, they can't even bring the iron back to the cell. Like, there's a physical barrier there preventing that from happening. So you're stopping the E. coli from stealing iron from the host where it wants to, down in the crypts of the intestine. So now it can't colonize. And what's really cool about this technology is there are, in general, about 15 to 18 recognized different types of SRP proteins, but we don't have to get them all. If you poke enough holes in a boat, is it going to sink? Exactly, right? So you poke one hole in a boat, probably might be able to tread enough water to keep going. But if we can identify enough different types of SRPs in a population of pigs, or what E. coli's they carry, we don't have to grab them all. We just have to get enough of them to poke enough holes to interrupt this process. Okay. Next slide, please. So without iron, so essentially looking at this bacteria here, we're just going to say, for argument's sake, we've identified every SRP that this particular E. coli can carry. In the research that we've done, like I was saying, there are approximately 15 to 18 of these different SRPs that can be identified. Most E. colis are going to carry 4 to 6 different ones. So again, we're very much focused on 4 to 6 different types that about 95 to 96% of all E. colis carry. So yeah, you're going to have your stray E. coli that might have some different SRPs, but they are very, very rare. So when we're able to look at a herd or a flow of pigs and identify, okay, these are the different types of E. colis, these are the different SRPs that the E. colis in this flow carry, we can actually create a vaccine that generates antibodies against all the different types of SRPs that we put in there. So if we put 3 in the vaccine, they'll generate against all 3. If we put 6 in the vaccine, they'll generate against all 6. So it's truly just what goes into the vaccine. So that vaccine then will create these antibodies, which then coat the pathogenic E. coli and prevent that iron acquisition from getting inside the cell. So as you watch this animation, you can see that bacteria, it's swimming, moving around. All of a sudden, it can't get iron anymore. It can't function. And now it's black and it dies before it can truly cause infection. Okay. Next slide, please. So how do you get this vaccine going? It's literally a conversation with your veterinarian. We need to identify the different types of E. colis or salmonellas that you have in your herd, which is literally as simple as taking a fecal swab. You can send in intestinal tissue or, necropsied pigs, but if we can get a fecal swab and understand what types of E. colis are circulating in the herd, we can create a vaccine against it. So what we do is get these swabs in. We identify from a culture, is there E. coli in these swabs? We can do that with salmonella. Sometimes for salmonella, it's a little easier to identify from tissues because salmonella doesn't tend to stay alive sometimes as well on slats or surfaces. So E. coli will grow really well. Sometimes it's better to get samples directly from the pig. But again, it's as simple as a fecal swab. And then we take it from there. Next slide. Once we get that fecal swab from you, we grow that bacteria, which again is typically going to be the E. coli in an iron deficient environment. So what this means is we want that E. coli in the cell culture to express those SRPs. So once we identify what SRPs are there, because we've pulled that out of the culture, we can take those SRPs, purify them. So it is not a cell wall vaccine. It's not an RNA vaccine. It's not a bacteria component vaccine. It's literally taking those proteins off of the cell wall of the E. coli and then putting them into an adjuvanted vaccine. So what's really neat about this technology in that regards as well is there's nothing else for the immune system to have to respond to. You're not putting in extra carbohydrates, or you've possibly heard of lipopolysaccharides or different things that come off of a cell wall. There's fats on a cell wall of certain bacteria. It's taking all of that away and just putting these proteins in there. So not only does the immune system only have one thing to focus on, but it can really focus on that to generate a good antibody response from the vaccine. Okay, next slide, please. So kind of already touched on this, that we put those SRPs in a bottle with an appropriate adjuvant, but want to highlight that this is an injectable vaccine. So this could be really, really different than what you're potentially used to, because a lot of E. coli vaccines are oral, and that's not a bad thing or not a wrong concept, in that E. coli is in the gut. So you'd want to give a vaccine orally to try to combat that. But if you think about giving an oral vaccine, that vaccine has to go in the mouth, exposed to saliva, where it starts to get digested. It's got to go into the stomach that has acid. It's got to make it all the way through the small intestine. So depending on how you're trying to do that, sometimes you can have some issues with getting it there. When you inject a vaccine, you truly get an antibody response. You're putting that into the blood system. The blood system is then recognizing this is not supposed to be here. That adjuvant acts as a signal, like, hey, I'm here, like, come find me, immune system. So you're making the immune system work to generate these antibodies, and get those then down into the gut. And your gut is one of the most blood perfused systems that you have. Why? Because when your gut is processing nutrients, where does it got to go? Into your blood system, so that it fuels your muscles, it fuels your brain, that's your transport system to get those nutrients around. So when you think through the process of an injectable vaccine to work on the gut, again, that blood perfusion is critical in how your body sees nutrients, absorbs nutrients, and spreads nutrients. Okay. Next slide, please. So one of the studies that I've done most recently with Vaxxinova is, okay, well, let's put SRP to the test. There haven't always been a lot of really good challenge models for E. coli, because pigs are exposed to E. coli pretty consistently. So you have to find a strain that is going to infect pigs, because it's something that a pig sees pretty commonly. So through Vaxxinova research and working with some industry partners, we were able to develop a an E. coli challenge model. Like, if I put this E. coli directly into the gut of the pig, will it cause them to get diarrhea? So we did all of that pre-work to make sure we had an established E. coli infection model. And then ended up getting a lot of pigs in that we were able to vaccinate while they were still in their lactation phase. So at 3 days of age, which is very common for pigs to go through processing, it's when they get their tails docked, they get their iron shot, the males get castrated. You're already picking up the pig at that point in its life. So it's a convenient time to give vaccines for that pig when it's young. Because this is a 2-dose product, it's a killed vaccine that needs to be injected. You need that, what we call priming vaccine, followed up by a booster vaccine. So that priming vaccine, like I was explaining, give it at processing, and then give that boostering vaccine at weaning. So for different farms, that's usually anywhere between 20 to 24 days of age. So for this study, we were able to work with a sow farm where we could go in and vaccinate a certain number of pigs, again, at 3 days of age and at 21 days of age. And then we also purchased pigs from this farm that we didn't vaccinate. So we put them in a barn that we could do challenges with. And so both of these pictures, as you can see, unvaccinated and vaccinated, same age of pigs, came from the same source, had all the same iron shots processing, Circo vaccines, all those core vaccines. Literally the only difference between these two groups of pigs is the vaccinated pigs got SRP. The unvaccinated pigs did not. They all got exposed to a pathogenic strain of E. coli approximately a week after they were weaned. And this is what those pigs look like 3 days later. So, I know you're all on mute, but
Wow.
you can answer
Wow.
in your head, which pigs would you rather walk into and see?
Wow.
Yeah, it was crazy. I mean, this wasn't even half as bad. Like, just manure everywhere, all over the walls, sprayed. I mean, yeah, it was like a combat zone in those rooms for a couple days. So, pretty crazy. Next slide, please. So, wanted to take a few moments to talk to you about some producer success stories because, sure, it's easy for me to say, hey, we have this vaccine. I've done my own research trials. Well, what have we seen in the hands of customers? So, we'll call it System A, is a part of the Pork Powerhouse 40. So, if you're not familiar with that list, there's a list that gets put out every year of, call it, the top 40 swine producers in the United States. Smithfield is always at the top. They have millions of pigs, right? And then you get down into, like JBS would be on that list. Passion for Pigs would be maybe more towards the bottom, but their producers might be there. If you've heard of, New Fashion Pork, Pipestone System, the Triumph Group, Christensen Farms, all these larger pork producers are some of the examples of who you find on this top 40 list. So this System A was having issues with E. coli control primarily after weaning. We had a big run of E. coli disease in pigs post-weaning here in the last few years, and trying to understand why all of a sudden are we seeing this? Is it feed related? Is it genetics related? Is it production related? How we're taking care of these. But this group has used approximately 280,000 doses of SRP vaccine to date. They've used it in pigs across multiple sow flows. So again, if you're not familiar with that term, sow flows would be this sow farm putting out weaned pigs compared to a different sow farm putting out weaned pigs compared to a different farm. So different sets of moms, different sets of exposures. A lot of times it's common that sow farms will commingle these pigs together. And that can sometimes be just a recipe for E. coli issues, because you're bringing animals together with a group that have never seen each other before, never been exposed to each other before. And that can cause some gut health problems. But they were experiencing a lot of death loss, up to 10% mortality in some of these farms. Adding SRP was able to take it down to 1%. They're now on, they should be even past their 8th group of pigs, but using quite a bit of this vaccine and seeing a lot of success with it. Next slide, please. System B is actually our longest using SRP customer. They actually started using SRP for the salmonella control and then added E. coli in 2024. They've used over 500,000 doses to date. So using the salmonella SRP was actually for their boar stud. We were seeing a lot of non-select, poor performing, poor semen quality from these boars that were experiencing severe salmonella issues, and was able to get that cleaned up and get a lot better performance out of those boars. When they added the E. coli later, it was actually for a sow farm that is an antibiotic free flow. So they don't have the ability to treat E. coli in those pigs with your typical gentamicin, ceftiofurs, your exceeds, your different antibiotics, because again, they couldn't use them. So not only have they used this in antibiotic-free pigs, but they've used it in what we call normal commercial pigs, used it in their boar stud. Saw really good success across different types of pig farms and pig flows. Next slide, please. System C came to us. They were actually seeing E. coli mortality in excess of 40% in their gilt developer. So again, gilts are the future of your farm, right? You're bringing them in, you use them as your replacement so that, you know, as your sows start to age, you can keep that herd productive and moving forward. So to lose 40% of your gilts, animals that you brought, animals that are supposed to be the future of your herd, can be really devastating to keeping a sow farm going. They had been using oral vaccines, lots and lots of feed and water medication, and just couldn't get this under control. So started using SRP in their gilt developer, and don't have a number on it from what it went down to 40%, but in their words, shut down mortality. So would like to think that, again, that's dropping down to that, you know, 1% type range that really helped them get control of their scour issues, E. coli death loss in very high value animals that sow farms need to function. Next slide, please. So as promised, I wanted to throw in a little bit of poultry and dairy data. So two of the main studies that Vaxxinova has done looking at SRP in E. coli and salmonella in broiler breeders using this vaccine has had many farms to go down to 0% mortality from E. coli, reducing colonization of E. coli, salmonella in the reproductive and internal organs. They've actually shown a duration of immunity in birds up to 62 weeks of age, which is just absolutely incredible that these animals can hold that on for so long. And they can find maternal antibodies in chicks out to 14 days of age, which again is critical to get those chicks going and started. Most of the SRP in the poultry world is actually given to the what I'd call multiplication hens. So not the hens that we would necessarily eat. They can pass that immunity on to their chicks so well that commercial birds are not vaccinated because they can hold on to those antibodies and keeps them protected for extended periods of time. So, you know, obviously birds lay eggs, right? It's a little different from a pig. Pigs are completely dependent on colostrum for antibodies. So this is something that we kind of need to investigate more with pigs to see how much maternal immunity is. But from our bird friends, we know that these antibodies can last a long time and maintain protection against challenge. Next slide, please. So for dairy farms, this vaccine has been key to help prevent Salmonella, Klebsiella infections. Salmonella more so for calves, seen a 99% reduction in fecal shedding and a 97% decrease in moderate to severe diarrhea in calves. And for those of you that work with calves, I mean, salmonella can be absolutely devastating to dairy or beef, you know, for that matter. Those calves just do not have the resilience to have diarrhea and lose all that water and come back from that. So it's been a huge help for salmonella control. Klebsiella is more important, you know, as a mastitis pathogen. When vaccinating cows, they saw a 77% reduction in mastitis, and decrease by 42% of those somatic cell counts, which you know is important for milk quality. And they actually saw in vaccinated cows increased milk production by either vaccinating for salmonella or Klebsiella. Just helps that animal have so much of a healthier gut, so those nutrients that they're eating go towards making milk and not fighting off disease. Next slide, please. So getting back to pigs, you know, for commercial hog production, if you could advance again so my data pops up. Thank you. We have made over 10 million doses of SRP to date. Again, going into sows, gilts, nursery pigs, boars. We only have autogenous Salmonella and E. coli for pigs, whereas the slides I was showing you previously for poultry and dairy, those are actually commercial vaccines. It's something that we would look into potentially make this as a commercial vaccine for pigs, but all autogenous up to this point. Next slide, please. So you've heard of other E. coli. Oh, yes, please ask me a question.
Andrea, when you were talking about the commercial vaccine versus an autogenous, explain what that difference is.
Oh, yeah, absolutely. So a commercial vaccine, another way to say that is off the shelf. So you, and those are going to be typically companies like a Boehringer Ingelheim, a Merck, Zoetis, Elanco, and they are going to be distributed through a veterinarian, but those are vaccines that they make in bulk. It's the same vaccine, call it bottle to bottle to bottle, that you go and purchase. So things like, you know, I'm obviously more familiar with pigs. So FarrowSure from Zoetis, your parvo, Erie, lepto vaccine. You go to your veterinarian, you say, I want a bottle of FarrowSure, you buy that off the shelf, okay? What autogenous vaccines are going to be geared more towards endemic pathogens that don't have an off-the-shelf option. So strep is one of them. Flu would be one. Flu changes so often. You know, I just gave Zoetis as example. They do have an off-the-shelf flu vaccine, but it is not going to be the specific vaccine that you have in your herd. So autogenous is, okay, I have a strep problem. You send us your specific strep. We turn around and make a vaccine for it. So in this example for SRP, you have a particular E. coli salmonella in your farm. You send us that isolate. We make you a vaccine specific to that isolate from your farm. Does that help? Awesome.
Yes. Yes.
So different E. coli options out there. And I, you know, don't necessarily want to spend a ton of time on this, but different vaccines will target different things. So if you look at what they call a core antigen vaccine, it actually targets the endotoxins that an E. coli produces once it's colonized. So if you're targeting an endotoxin, that E. coli's already been able to go in and start to cause damage and is releasing those toxins into the gut, core antigen vaccines are going to attack those toxins to help reduce clinical signs, but they're not going to stop infection. J5 vaccines will target the bacteria cell wall, but they're typically going after sugars or fats, which are not easy for the immune system to recognize. Oral vaccines, that's basically another way to say an outcompete vaccine. So the concept with those is that I'm just going to put a ton of really good E. coli into the intestine to get that to overgrow and colonize, so that any bad E. coli doesn't have the ability to get in there and cause infection. But that's highly dependent on timing. Like if you don't get that good E. coli in there before the bad E. coli, it's not going to help you very much. Whole cell bacterins, again, target the bacteria cell wall, but they don't always have the ability to cross protect, because again, they're going after sugars, which are not very species specific. Like different E. coli's can have different LPS or sugars on their cell wall. Whole cell bacterins, on a whole nother note, are very good for streps, because strep, or what's now referred to as Glasserella parasuis, if you're a little older like me, you learned it as Haemophilus parasuis. Yep. You want to target those whole cell with whole cell bacterin, because those are very specific capsules. So kind of show this to show that the different options are there, but different antigens, different vaccines that work against different things are good for different types of bugs. But what we want to demonstrate here is that, again, when you use SRP, it's going after that protein that's conserved. So you're getting the best of a lot of different worlds, because then it's targeting in a protein that it can recognize. It's making a really good immune response, and it's getting that bacteria before it can cause infection, because it starves it while it's trying to cause infection. Okay. Last slide, please. So kind of already went through a bit of a summary, but what I really want to make sure I point out is this technology only works for gram-negative bacteria. Strep, GPS, Bordetella, or actually Bordetella and Pasteurella are gram-negatives. I shouldn't actually go there. Why am I blanking on other gram-positive bacterias? Like mycoplasmas. This technology doesn't work for every type of bacteria. They have to be in the gram-negative category. It is a 2-dose injectable. It's, again, very important to have that priming and then boosting of the immune system, which then gives you that strong memory response because you've got antibodies on board. I've actually done studies where when I vaccinated pigs twice, and then when I challenge them, you actually get an immune boost from challenge. It's like them getting exposed to E. coli makes the immune system go, oh yeah, I don't like you, right? I'm going to make more antibodies. So it's another benefit of having this be an antibody vaccine with that strong response. Blocks that iron, kills the bacteria. We're not going after the toxin. We're not trying to outcompete. We're outright killing the bacteria. And just here recently, our company's been able to get approval through the USDA, which I want to make sure I make another point of, as Vaxxinova is a USDA approved and monitored facility. We have standards that we have to follow. We have regulations for quality and control. You know, we're not just throwing antigens in a bunch of water and then saying, here's your vaccine. Like we have to go through certain quality standards. So in that approval process of how we make SRP, we've now been able to get approval through the USDA that we can combine this SRP technology with what we call autogenous whole cell killed. So if you want to make a combo vaccine that if your flow suffers from, say, Salmonella, E. coli, and then again, like strep and mycoplasma are pretty common ones, we can mix those all together to give you a one-shot vaccine, because nobody likes to give pigs multiple shots. Pigs don't like to get multiple shots, for that matter. So it does add that convenience factor as well. So with that, that's all I have. And my last slide is, what questions do you have for me?
So I've got a question. If we're talking about autogenous, so we're making it to the specific bacteria at the farm, how long, what's your lead time on that?
Yeah, that's a very good question. So once we get an E. coli in, E. coli grows pretty well. We've got that E. coli grown over a couple days. We like to do what we call whole genome sequencing on it because I want to know what type of E. coli it is. What virulence factors does it possibly carry? We can tell that from doing a sequence of the genome. To go through the process of growing the SRP in an iron deficient environment, understanding what SRPs that bacteria has can take approximately another week or so. But once we talk with a client or a producer, the vaccine can go into production. And if I had no other vaccine to produce, would maybe take 6 to 12 weeks. Unfortunately, we have a lot of customers that want SRPs. So to get into the queue, sometimes we are looking at 4 to 5 months to get vaccine out the door. Now, all that said, we can work through a USDA process called non-adjacents. And say we already have an SRP vaccine available, if we can work with your state vet to say, look, I found this E. coli in my herd. It's a match to an E. coli found in another herd. This is how they're potentially connected. We can get vaccine into your hands a little bit faster. That wouldn't be your specific isolate. But with this technology, it doesn't have to be. We just have to make sure those SRPs match up because, again, how conserved they are across different bacterias. So we have a couple different routes. If you want your own vaccine, it does take a little bit longer to grow it and have it within the queue. If we can get non-adjacent approval through your state vet, we can get you vaccine within a couple weeks.
Excellent. Thank you.
Anna, did you have anything to add to that? I should introduce my colleague, Anna Guess, is on the call. She is my main account manager. And so she's way more savvy with those things than I am.
I would have to say you covered the bases really well. Yeah, it's, I mean, obviously, to Andrea's point, we always want to make sure that we're doing our due diligence and ensuring that there's going to be adequate coverage there, and making sure that the state veterinarians and the herd veterinarians are all involved in that process. So we do make sure that the communication is clear and consistent from that point of view.
Gotcha. Good to know.
So, a common question. Oh, go ahead.
So what's the, you do all this work, right? It takes 4 to 5 months. What is the point at which it pays for itself? How many doses do I need to order or do I have to have in order for the cost benefit to be there? Does that make sense? Do you see what I'm asking?
Yeah, no, I know exactly what you're talking about because we don't want to act like pigs or cows or chickens are widgets, but that they are a part of our supply chain to create food. So yes, we want to have efficiencies. We want to see a return on investment, for what we're putting into these animals and producing safe and good protein for people to eat. So the struggle in answering that question for you relies more in how many doses that you initially make. And I hate sounding like a vaccine salesman, but the more doses I can sell you, the cheaper they are, because when you produce things in bulk, that just makes that process more efficient. So actually, I'm going to reach out to Anna again to answer this question. I don't know that we've completely calculated that break even, because we have one customer that ordered 10 million doses, and we have another customer that'll order 50,000. So it's a little difficult. Anna, can you pop in and help me with that one a bit?
Yeah, essentially, if we are able to save two pigs, the bottle pays for itself. Like that's the mark that we're looking at. The technology, when you compare it to other technologies, can be considered more expensive when you look at one particular widget. So if you just look at oral vaccinations, if you just look at autogenous vaccinations, if you're vaccinating your sows to get that maternal antibody immunity to those pigs, are you vaccinating those pigs? Do you have an antibiotic program? There's so many different components that go into a vet med program that we're still hashing out from an ROI perspective. But what I can say is when we see certain flows, when we see barns that are erupting and catastrophic hemorrhagic E. coli, if we can save two pigs, there's your break even.
Okay. So do you guys have a minimum number of doses that you have to order? How big do you have to be in order to utilize this technology?
Sure. So with the appropriate non-adjacent paperwork, there's essentially no minimum that you have to meet. If you have to go through your own autogenous route, they do require a minimum of about 10 to 20,000 doses.
And that includes the first dose and the second dose.
Yes, ma'am.
Yep, that's just total doses ordered. So yeah, you would want to order for your herd size, calculating, I want to give 2 doses per wean pig. We're just starting to see some farms look at, well, do I want to do 2 doses to my sows pre-farrow if I've got lactation scour issues in addition to post-weaning scour issues? So you could be looking at 2 doses per sow, then 2 doses per wean pig, but the majority of animals that we see is 2 doses per wean pig.
And those are going to be the same whether we're doing the sows and the post-weaning.
Correct, because you're looking at it as a flow, right? I mean, your sows are birthing the pigs that are going into your wean to finish system. So everything you've heard about F18 E. coli in the nursery versus K88 versus K99 E. coli in pigs that are still nursing, throw that out the window, because with this technology, it doesn't care if you're a K88 or an F18. It's going after those surface proteins, which a K88 and an F18
Are the
could
same.
both carry. Yes, exactly. Exactly.
Okay.
So as long as the diagnostics that you submit to us, if you'd submit us E. coli that you find from your nursing pigs or pigs that are still on mama, and you're also sending us E. coli that you find in your wean to finish system, we could combine all of those into one product to give you that diversity of SRPs in the bottle. So you could give it to sows and you could give it to piglets. You could give it to your boars, you could give it to your incoming gilts. It's just trying to find all those different SRPs that you have across your entire flow.
Gotcha.
That certainly makes things easier if I don't have to worry about which one of these goes to mamas, which one of these goes to babies. If it's just a singular product.
Yep. Well, and like I was telling about strep, if you're going to make a strep vaccine to cover your system, like we have some farms flows that have 4 or 5 different streps in the same bottle because they found those. Like I have to have each one of those streps, each one of those Glasserella parasuises, because it's just a different way of targeting a vaccine. This technology, I don't have to have that, because an E. coli that you find in the nursery could carry the same SRPs that you find in your nursing pigs. That's the beauty of the conserved process, or part of how this vaccine works. It's going to target that protein, whether it came from a K88 or an F18, an F4 or a K99, nursery or finisher or lactation.
Fantastic.
Yeah.
I love this. It like gets my geek on, like.
Oh, have you been listening to me? I've been like geeking out pretty much the whole hour.
I love this stuff.
Fantastic. Well, so I do want to thank you for giving us the presentation tonight, for explaining to us what SRPs are and why it is a good target for a vaccine, why it's something that potentially we're going to see more and more of coming in the future. Just those 2 pictures, if it can take those pigs that are covered in manure because the diarrhea is everywhere to a group of pigs that just looks like they feel so much better. Just the welfare implications of that are fantastic.
Absolutely.
Let alone the monetary, I didn't lose pigs.
Mm-hmm.
Right.
Yep. Yep.
So that is fantastic. That is fantastic. Thank you so much for presenting to us tonight. I'm not trying to cut off any questions, but I did want to throw up, we have our survey for, how do we do tonight? Did we answer some of the questions that you had? Are there other things that you want to be learning about? So we have both a QR code and or a link that you can follow. The link is also dropped in the chat, so you can go ahead and click on that if you'd prefer to. If you have a separate phone that you're not using to be online right now, you can take a picture of the QR code and follow that way. We're very excited about our upcoming sessions. Just a reminder, and we have recorded this session tonight, so we will have it available on YouTube in our Pearls YouTube. So this is not necessarily available to everyone. But it'll be available through Pearls after we've had our in-person presentation. Thank you again, Dr. Pitkin, for talking to us tonight about SRPs. Do we have any other questions? Well, we still have people here tonight.
And thank you, Anna, for joining us as well. Anna will be the speaker on November 8th. So you'll get to meet her in person.
Yes, thank you guys.
Looking forward.
Super exciting.
Yes. I couldn't do this without Anna. And just it's a neat thing about Vaxxinova that we try to have the veterinarians be veterinarians. Isolates come in, help do the diagnostics, give you that formulation advice. Once the vaccine's formulated, you don't want my expertise anymore. You want your account manager. Anna works hard with our customer service, maintaining inventory, understanding, reorders, making sure shipping happens. It's always a team effort to make sure that we help the customers out. So want to make sure to highlight how important Anna is in her role and in the vaccine supply chain.
Sounds like it's a
Thank you.
team effort.
Very much so.
Fantastic. Well, it is 8:10. I want to thank everybody for attending tonight, for learning more about SRP technology, as well as, sorry, I'm trying to eradicate myself over here, but as well as thank Dr. Pitkin, as well as Anna, for giving us this presentation and letting us learn more about it. We are excited to be ready for our upcoming sessions. Our next session we have is Pixels of Production on October 28th, where we'll learn more about our Heroes to Hives, and then our in-person session coming up on November 8th. So make sure to keep in touch with us, follow us on Facebook, keep an eye out for our website, and we will be sending emails to see how everybody was doing this weekend. So with that, I'm going to go ahead and close this down. But again, thank you very much for attending tonight. Thank you very much for your presentations. We learned a lot.
Good, good.
Thank
Please
you. Thank you.
questions. Always happy to help.
Excellent. Thank you very much. All right. Enjoy the rest of your night, guys.
Good night. Thank you.
Good night.
Right.
Bye.